Welcome to the Web EV Talk series. Today is the 26th May 2020 in Australia. And it's my pleasure to introduce Ana Claudia Torres Silhas. She's an associate professor at Federal University of Sao Paulo, Brazil. She's also an ISEF board member, members at Large America. So Ana is an expert in immunology and molecular parasitology and host-parasite interactions and EV and pathogens. So she has worked in the fields for more than 20 years. Her research papers covered the characterization of the cellular and immunology that control the interaction and modulation of protozoa parasite. Ana has established some basic principles of Trypanosoma cruzi interaction with host cells via extracellular vesicles. So today she's going to present her talk on extracellular vesicles in protozoan infections, host cell communication. Welcome, Ana.
Okay, and I would like to thank for Carolina, Dolores, and Ian for the invitation for talk my research. I worked for more than 20 years in characterization with EVs in protozoa parasite. My talk today is extracellular vesicles in protozoa infections and show how they interact to parasite in the whole cells. And for a start, I need to figure out what happened this tropical neglect tropical diseases. 1 billion people suffer for one or more neglect tropical diseases in Latin America and South Central America. It's a big problem of health in the Americas. And neglect tropical diseases, you have a lot of example for protozoa like Chagas disease. This is my research, my point. Leishmaniasis, human African trypanosomiasis. In the virus, you have a lot of neglect tropical disease like dengue or chikungunya, and the bacteria, leprosis. For the helminths, the parasites, you find a lot because is neglect tropical diseases is a diseases to poor people and the bad conditions in the 149 countries affect 1 or more billion people and cost a lot of billions for economy. And in the last November, I went to the Institute Pasteur to discuss about the future of tropical neglect diseases. And you have a lot of diseases to attention for discover the compounds to treat or vaccination, but still a lot of questions about the parasite interaction with the whole cells of the host. When the questions about the Chagas disease, the drugs to treat it is a lot of problem to affect the human, but you need to development a lot of preclinical compounds to find the vaccination, and the Chagas disease is the goal now. What about the disease? Is the Chagas disease or American trypanosomiasis is discovered a long time ago, as you can see here, in the paleoparasitologist recovery Trypanosoma cruzi, a long time ago in the mummies in Chile. And this is a Dr. Carlos Justiniano Ribeiro Chagas. His discovery, this parasite, the forms of parasite, the life cycle, and the phase of the disease in Brazil a long time ago too. And in 1972 to describe these compounds to destroy the parasite, but has a lot of effect when treated. And the science published the completely genome of the Trypanosoma cruzi is good because is a line to found the new compounds to treat the Chagas disease. And the Chagas disease is transmitted for the vectorial transmit for the Triatom infestans, affect the 8 million people around the world. You find the 2 drugs to treat the Chagas disease, and that a lot of people die to complications for the chronic or a chronic phase in the Chagas. And the WHO consider is a neglect tropical disease, but no or little interacts to big pharmacology company to development two drugs to Chagas disease. Is a big problem for us. For the transmission in the endemic area in the Latin America, 8 to 10 million people in fact here in the Central America and the Latin America, but in the North America it spread a lot of infection. When the big problem increased the immigration from endemic countries and affect North America, Europe, Japan, as well as Australia. But the blood banks in Latin America control the transmission of Chagas diseases. And the people move to countries like America and not check the blood banks, and the transfusion is a transmission for the diseases. And you find now a lot of vectors, triatoma infections to move in the south of US and transmit this disease for the people in America. For the transmission is 8% is the Triatoma infestans, a lot of species to the family, to the vector to transmit these diseases. The north in Brazil will have a lot of oral transmission, the 10% for the contamination food like or sugar cane or the acai food or the guava juice or the orange juice transmit too. The congenitally, the blood bank is a big difference because need to check the antibody against the parasite, organ transplant, and few cases for the lab accidents to Chagas diseases. But the life cycle, I don't put a lot of energy to explain the life cycle. The life cycle is very complex, but the vector, the vector contains the proliferative forms to epimastigote, and the vector, take blood meal to ingest this, and when it finish in the feces contain the metacyclic tripomastigotes forms. These forms is in the intracellular parasite invasion cells, and proliferation transform these replicative forms as mastigotes in the human, and broke cells, and the more parasite invasion, more cells. This form, the parasite in human, my lab, my goal and my research work to tripomastigotes forms. These forms, I try to understand our questions to host parasite interaction, how the parasite modulate the immune system, how the parasite invasion these cells, and our work is not in the vector phase, is the parasite form, but the tripomastigotes forms. And the Chagas disease contains two phases, the acute phase and the chronic phase. This is 4 8 weeks, and you find a lot of parasite in the blood. After that, the move for the organs. In the chronic phase, you have IgG, a lot of the subpatent parasitemia, and increase the immunoglobulin, the IgG. Is the chronic phase. It's a big problem, and I have a lot of problem to heart and the intestine, the people. The people lose the function to the megacolon, megaesophagus, the heart is increased, and the people lose a lot of quality to health, and lose is an impact economy when a lot of people infection to Chagas disease. And what about the parasite, the Trypanosoma cruzi? This is a scan electronic microscopy to show how the morphology to the tripomastigotes forms. And I would like to start to show how the major molecules, 6 and 8% of the parasite contain glycoconjugates like mucins, or contain sialic acid to the polypeptide core here, transalidase, GP85 glycoproteins, and glycosyl inositol phospholipid. And this control the parasite interactions, parasite invasion, and induce the human immune system. It's very important to pay attention to this molecule to express in the plasma membrane in the parasite. For the mucins, this molecule is glycopeptide. It's very important because contain alpha-gal epitopes. These epitopes, the human, when infected with the parasite, produce lytic antibody against this epitope. And this epitope is very important because you have a hypergamma-globulinemia and started to humoral immune response. It's very important, this molecule, to control this parasite interaction. And the parasite, the glycoconjugate induce a host immune response, induce cellular immune response and the humoral immune response. In the innate immune response, you induce activation of the macrophage, activation the natural killers, and after to move to produce a lot of antibody, produce pro-inflammatory cytokine. On the other hand, the humoral immune response produces lytic antibody against alpha-gal epitopes. It's very important to pay attention to these questions. But 25 years ago, I have a lot of questions. The parasite has a lot of glycoconjugate, but how this glycoconjugate interact with host cells? These mucins expressed in the parasite surface interact direct to the plasma membrane, as a lot of co-receptor, induce the innate immune response for toll-like receptors. Very important to induce the innate immune response. The signaling is induce the MAP kinase, translocate NF-kappa B, and induce the immune response genes and produce the cytokines. But you have the two alternatives. One is release soluble molecules, and one you find in the vesicles. Is a 25 years ago, a lot of questions about these interactions, and they moved to understand this. And are pathogens shed in vesicles? Is a big question about this. How molecules interact with host cells? Would these molecules release from the pathogens, and which forms? You have a lot of examples to leishmania. You find the fungi, you find the bacteria, virus, modulate the interaction of the host cells. And for the Trypanosoma cruzi, I show our results to convincing it's very important to vesicles to modulate the parasite whole interaction and communication. In the first evidence to see the triple mastigotes forms, it's not the other forms of the parasite, because it's a long time ago, the same group showed this epimastigotes forms release vesicles, is the plasma membrane, the name is 30 years ago. The Dr. Walter Colley and Maria Julia showed the first time these triple mastigotes release vesicles. Is the first paper in the protozoa parasite in the triple mastigotes form release vesicles. For this work is very important. And here, as you can see, is the evidence we publish in the FAPESP news. This is in gray is the epithelial cells. In red, the triple mastigotes form is a scan electronic microscopy. As you can see here in yellow, the vesicles for all parasite release vesicles and is still alive. And you have here is the electronic macroscopy and the triple mastgold's form. We published with my Dr. Walter Colin Maria Julia in my first postdoc. Triple mastgold derived to infected mammalian cells release extracellular vesicles. And the size is a 50 to 100 nanometers. And here's the, as you can see here, is the arrows is a vesicles to release, and this is a vesicles in the ultracentrifusion to see this better characterization. And the scan electronic macroscopy, again, the triple mastgold's form release vesicles, as you can see here. And these are published is a review for more example for triple mastgold's form release, the convince to release vesicles, and don't exchange the morphology to release these particles. And how fractionation and characterization. I started several years ago to use the Cepharose CB4B to separate the serum, the molecules to serum and the EVs, and I collect the parasite in supernatant and add the samples in this column, and separation for the fractions, and this is a very rich fractions, and content is a smear is very characteristic to mucins and TC85 molecules. In the follow to use the antibody purification against the chronic patient with Chagas disease and contain alpha-gal epitopes. The alpha-gal epitopes, if you remember, is still in these mucins in the glycoconjugate surface of the parasite. For the questions, we publish about this, the flow cytometry analysis of Trypanosoma cruzi, the viability after the shedding procedures is still alive after 3 hours for shedding. 99% the parasite is still alive. Is a spontaneous release, is a process, is very spontaneous. But the first question is these EVs contain glycoconjugate like mucins and responsible for the activation. For the proteomic analysis, my student Kleber Ribeiro, we published in journal Extracellular Vesicles in 2018, showed the 6% of the hits, the proteins, we found the transalidase, GP85, mucins, and cruzipine. And it's very important because the mucins represent the plasma membrane of the parasite. It's very important result. And you confirm if you, as you can see here, to Western blot, and you use the, my group used the antibody against for parasite membrane, and Y and UU, different strands, express different concentrations of the glycoconjugates. As you can see here is like mucins, transalidase express, and cruzipine is contained in the vesicles released from triple mastgold's form of Trypanosoma cruzi. In parallel, we started to work to EVs to see if the EVs promote the interactions and probably internalization the whole cells. As you can see here, the experiment, my students label cells with the P8K26 to label the vesicles, and after incubation 3 hours and A to endothelial cells, 3 hours, and wash this in vitro interaction, and 24 hours. As you can see here, these dots, the pink dots, a lot of vesicles and the cytoplasm of endothelial cells. And do EVs, parasite, release these EVs, trigger different immune response, what the receptor or no receptor interaction, what pathway are involved. For the reason to see the toll-like receptors, my ex-supervisor for my PhD, Igor Correia de Almeida, and Dr. Ricardo Gazzinelli showed the mucins isolated from the parasite interact toll-like receptor. And we start to see these EVs interact to toll-like receptor. For this, we isolate the peritoneal macrophage from the wild-type mice, and for knockout mice from toll-like receptor 2. And after 1 week, collect the peritoneal macrophage and incubation of extracellular vesicle from Trypanosoma cruzi and isolate from quaternion. After many days, collect the supernatant to see nitric oxide and cytokines, pro and anti-inflammatory cytokines, to see if these EVs induce or and depend for this innate immune response receptor. As you can see here, you published this article in 2015 in Journal Extracellular Vesicles, and Paula Nogueira showed one incubation in macrophage isolated from wild-type mice and knockout mice. You can see here is when we added the 2 different concentration of EV from parasite and contain a lot of controls here. As you can see here for TNF alpha to abolish completely one incubation, the macrophage isolate from knockout mice. And EVs from triple mastigotes does not activate macrophage of toll-like receptor knockout mice. Depends the toll-like receptor to induce the human or mice immune response. And we have the pro-inflammatory response and abolish completely when you don't have toll-like receptor. But you have a lot of questions. If you incubation with EV isolation from parasite, what happened this macrophage? As you can see here, incubation for parasite and EVs from isolate from parasite in human macrophage, T8P1 cells. As you can see here, 1 hour, 6 and 24 hours after incubation is a scan electronic microscopy. You see the plasma membrane of this human macrophage, a lot of vesicles. And they start to characterization. The parasite release vesicles. These vesicles induce the macrophage to release more vesicles in the micro environment and induce the pro-inflammatory response. Why? Because the parasite is an intracellular parasite. It's very important to parasite to spread infection more. And as you can see here, the characterization for a nano-NTA. You find here is the markers to EVs from isolate from macrophage. And when you infect cells, the macrophage release more vesicles than control without infection. And we'll have you in parallel start to see how many particles are needed to activation this toll-like receptor or toll-like receptor 4. For the toll-like receptor, you have a lot of molecules from isolate from parasite EVs to induce this interaction. I use here the C8O cells transfect toll-like receptor 2 or 4. Depend this interaction to use the interaction to the EVs isolate from parasite or macrophage, the toll-like receptor. And when I incubation this human macrophage with EVs isolate from parasite, as you can see in the heat map, in 1 hour or 24 hours, you find a lot of these studies of vesicles, pro-inflammatory hits you find here. And in conclusion, the first part, the parasite release vesicles. These vesicles interact with the host cells, induce the pro-inflammatory response, and these EVs and the parasite induce macrophage release more vesicles. And this microenvironment induce the parasite more invasion. For the next questions, these EVs promote or not invasion by triple mastigotes. As you can see here, is epithelial cells, the parasite invasion, endothelial and epithelial cells, pre-treated with 1 hour with EVs, after wash and infect with the triple mastigotes. After 18 hours, wash and label and count the number of infected cells. As you can see here, when you pre-treat these host cells with EVs isolate from parasite, you induce more infected cells. And these EVs is promote an increased invasion by triple mastigotes. For the last experiment about the invasion, I use the same cells, CHO transfected toll-like receptor 2 or 4-2C. It depends this receptor to increase more invasion. Yes, I use the total EVs isolate from parasite and EVs isolate from SEC. And I use 2 different concentration of EV measured to NTA, and pre-treat the cells, the same approach the experiment, and after infection and after 18 hours, fix and count the number of infected cells. As you can see here, is dependent for toll-like receptor to increase more invasion. And what kind of molecule modulate the parasite invasion? If you remember, in the first part of the introduction for the plasma membrane of the parasite, contain the alpha-gal epitopes. And we treat these EVs with the alpha-galactose enzyme and repeat the same experiment. As you can see, you reduce invasion, and this experiment is very important to see. One hand, the cells express toll-like receptor, and the other hand, the parasite, they contain these mucins, alpha-gal epitopes. It depends to enhance the whole cell's invasion, these molecules like alpha-gal. And long time ago, I published in the in vitro part, I finished and I moved to animal pilot, in vivo model. And we treat the animal with EV, and after infection, and see if you increase the parasitemia in the animal model to Chagas disease. We have in my lab the model to acute phase for Chagas disease. And after, to see what happened in the heart, in the other organs, what happened, the infection, to examine the effect of trypanosoma vesicle during the animal infection. And they treated the animal for a week with EVs, and after infect the bloodstream triple mastigote, IP, and I see parasitemia, mortality, and an histopathology analysis after 15 days post-infection. As you can see here is when I treated the animals with EVs isolated from triple mastigotes form, increase the parasitemia than control. But what happened, I'm shocked, because when I treat with EVs, the animal die before the control. But I look to histopathology to see what happened. And we'll find in the heart a lot of mastigotes. When you treat the animal, you find more inflammatory response, and this inflammatory response is pro-inflammatory response, and after days, the infection in animal is modulate from the pro-inflammatory to anti-inflammatory response, and the animal starts to die. And my group moved and to understand if EV biomarkers detect in the application clinical diagnostics. And my students, the PhD, Rafael Madeira, started to collect samples for chronic patient with Chagas disease and started to characterization the EVs if I found the biomarkers, or is EVs is very important to modulate the human immune response. And this is, we have a lot of chronic patients in the University of São Paulo. We have a collaboration in the people to work to chronic Chagas disease. And my students, Rafael, collect a lot of samples, separated, follow to MISEV to separate these EVs from plasma from chronic Chagas patients, and the control without Chagas disease is this, the balanced weight and the ethnical and the female and men. And as you can see here, the chronic patients is the concentration of particles is decreased than control. And Rafael started the immune assays to see, he used the human macrophage and incubation with EV isolation from chronic patients to Chagas disease and controls. And after 24 hours and 8 hours, collect the supernatant and see the cytokines like interferon gamma and IL-17. As you can see here in the chronic patients, the EVs induce the human macrophage more interferon gamma and IL-17. Is a preliminary results, but it's very interesting. And Rafael moved to more experiment to understand and follow these patients to see if you found the difference in the chronic patients. And in conclusion, this EVs isolate from parasite is very important because it interacts the host cells, immunomodulation and the activation of the pathogens, maybe is a prophylactic and therapeutic to use in the futures, or not vaccinations and markers to diagnostics. You found a lot of virulence factors like mucins, like TCH5, and this conclusion, this parasite, our big question is, the parasite release vesicles? Yes. And these vesicles contain the glycoconjugates, is the major glycoconjugates, and interaction toll-like receptor, and induce the pro-inflammatory response. And these activations induce the MAP kinase. We have a lot of, Paula Nogueira published the MAP kinase pathway and induce the cytokines and chemokines. And the pretreated cells with EVs, the parasite release these EVs and induce increase the invasion, because the parasite is an intracellular parasite. And these vesicles is control the parasite-host interaction. And I would like to thank my students, Kleber Ribeiro, he finished PhD, Camila Yoshida, Paula, Rafael, Lucas, and my colleague Patricia from Unifesp, Dr. Rodrigues and Dr. Paula Nogueira from Fiocruz, and the Weizmann Institute of Science, IFAT, Oriane, Matia Neta Regev Rudinsky, and UTEP, Dr. Igor Correia de Almeida, to help us during the proteomic analysis. And I'd like to thank the agency in Brazil, the federal agency in the CAPES, CNPq, and FAPESP, our group, Rafael, he worked in the patients to chronic Chagas diseases. Kleber worked with the proteomic and compare different strands. André published this year in the Frontiers about the interaction this toll-like receptor and a macrophage to induce, and Patricia and Rodrigues. And I would like to thank your attention, and I'm happy to take your question. Thank you.
Thank you, Anna. That was great. Let's start with the question from Kalyani, and then I have a couple of questions. Kalyani?
Yes. Thank you, Anna. I think this is a much needed work, especially in Brazil because of the tropical rainforest. So I have two questions. The first is that it is, of course, both of them are related to the EV release. So the first is that do you think that the release of EVs provide any advantages to the tree cruzi, like providing some drug resistance? And the second is that in the chronic phase of the Chagas disease, the heart and intestines are predominantly affected. So do also EVs show organotropism?
Okay. And the first, I publish not in EVs, but this you have a lot of strengths to resistance for drug.
Correct.
Very good questions for you. And I published this paper. If you send it for my mail, I send for this paper.
Oh,
I show the difference for the strengths, but a lot of resistance for the benzimidazole. But I never see yet these vesicles, what happened, maybe is moved to understand this drug resistance, because the Trypanosoma cruzi has a lot of strengths. A lot. Is a strength the DT1 and 6. It depends for the vector, depends for the country, depends a lot of, and I don't have yet to work to compare the difference for if the drug resistance decrease or increase the vesicles, the parasite release vesicles. And the second question, the tropism. When I infect the Y strand, I saw a mast goat's nest in the heart and intestine.
Okay. Thank you.
Welcome.
So my first question, so you showed very nicely when you labeled with PKH the vesicles that they are internalized, but we know the limitation of this approach because you can see many vesicles which could be broken vesicles or even the dye itself. I'm wondering if you have tried or if you're thinking to try to engineer cells to express the fluorescent mucins to understand if they are really the cargo that is inducing this reaction.
Yes, now I start to collaboration with Neta Regev-Rudinsky, and I label EVs from parasite with DNA, RNA, and lipids for specific, and I move it to interaction to image stream. Is a in vivo assay.
Okay.
In vivo cell. And to see this interaction. And I have a preliminary results to this approach, because this is labeling with sigma is not a good work for us, but it's not a good approach. I need to double check for another approach to see if the cells engulf these vesicles and modulate the environment into the cytoplasm, the host cells. And I move for the image stream.
That's great. And my other question was, what about the macrophages? The the THP1 are human, but they are from leukemia. Do you have access? Can you isolate macrophages from normal blood and
Yes.
have primary?
Yes. And Rafael, my students from PhD, started to move to PMPC to see if you found the difference on the incubation to EV to parasite and EV from isolate from chronic patients to see if the difference to induction is easy for us obtain the macrophage, peritoneal macrophage, monocytes.
Monocytes, yeah. Okay, the next question is from Ernesto Nakayasu.
Ah, Ernesto, my friend.
Knows him.
Oh, good to
Hello.
see you, Anna. No, my question is, again, I don't know if you have the answer, but when you show the data of cytokine production by the vesicles from donors, you know, the vesicles from the Chagas people seems to be like two groups. You know, there's like some that produce more, some that produce less. And I was just wondering if you have any kind of correlation with, you know, one the group say it has a better control of the disease, the other group has a worse control or something like that.
Yes, but the Rafael, when I saw this difference, the Rafael put the age, if the female, the man, one started treated, because some people in the chronic phase is started to treat the other drugs to see if you have a lot of problems. And now the folks in the Rafael to use this experiment to talk to Dolores, and now to move it to understand how these patients, what happened, the aid is induced or not, how many times did the first positive ELISA to Chagas diseases, how the treatment, if you the exchange for any drug to during the phase, chronic phase, if you is different to modulate the EVs in the plasma patients. And thank for your question, because Rafa started to look this point.
Yeah. Another quick question. So can you see alpha galactose in these vesicles from the patient?
Not yet.
Okay.
But is a good approach, because not yet.
Okay.
Thank you for this
well,
question.
No, I'll wait for it.
Okay.
Next question is from Carolina.
Hi, Anna. Great talk, and thanks so much for keeping it in time. So I have a question here, probably a bit naive. Have you looked into the heterogeneity of these vesicles that is being released by Trypanosoma cruzi? And is there any difference when they are by themselves and when they're infecting other cells or macrophage in the co-culture? Will they have like release different profile of vesicles?
It depends. This profile from just if you look the parasite and the strength is the same molecules, but if you look the virulence, parasite release more like transalidase, it's GP85, but if you look at the parasite isolated from blood, you find the same molecules, but more than in vitro parasite. Because virulent.
Okay, are they sort of like always smaller size vesicles? Like, do you see any like bigger size, like ectosomes size range, or because it looks like mostly like small vesicles, isn't it? Like
yes, I use the FFF. And I found the small population, but it's a preliminary result, a small population, and is like a exomery from the Linden group show, but it's the parasites like, and the exosomes or ectosomes, you find this population, but it's a 35 nanometers until 200 nanometers.
Okay. And my second question is like, is there a specific marker for these T. cruzi extracellular vesicles?
Yes, if you remember when I saw the mucins, you find a lot alpha-gal epitopes. When the patient start infection, produce antibody against lytic antibody against the alpha-gal epitopes, and the blood banks. I collect this contaminate positive for Chagas and isolate this antibody against. And the vesicles, this marker for us is completely different for the human cells is alpha-gal.
Oh, that's awesome. Thank you.
Yeah. Okay, thanks so much.
So there is one last question here from Alvaro Acosta Serrano.
Alvaro.
Alvaro.
Okay, I unmute him.
Hi, Anna. Can you hear me?
Yes.
Right. Okay.
Yes.
Hi. Okay. Great talk. And
Thank
I was
you,
curious about the release of EVs in blood, in whether an infection in animals or humans. Have anyone looked into the potential enhancement of infections when a tracheotomy feeds on infected blood, whether those extracellular vesicles may facilitate establishment of the parasite infection in the vector or not? And then the second thing is about, I believe also the vector forms may release some of these vesicles, and they won't have alpha-gal. So what are the markers? How can you identify them? Thanks.
Okay, maybe translocase, Álvaro. Do you see it? Because when you started to talk, I couldn't see. You ask about the vector or the animal model?
Okay, so the first question is in relation to whether the EVs release from blood trypanomastocytes may enhance parasite establishment within the tracheotomy vector. And the second is if the EVs from vector forms, what are the markers that you're using because they don't express alpha-gal?
No express. I published with Rodrigo Soares, with the PhD students of Rodrigo. In the EVs, you mill the vector Triatom infestans erroneous with EVs isolated from epimastogotes forms.
Okay.
And I saw the interference to development of the parasite. I published last year with Paranaíba and Soares. But the markers from the glycoconjugates is monoclonal antibody against glycoconjugates 35 and 50. You have the antibody against this. And Nobuki Yoshida sent for us. And I identified this marker for epimastogotes in the vector. And these EVs, your first question is very good questions, but they enhance a lot of glycoconjugates, but I don't yet to conclude this experiment for animal model. But in the vector is very important because release vectors, but the epimastogotes don't intracellular parasite, but the markers is completely different. And we have from the monoclonal antibody from Nobuko Yoshida lab to follow the expression in EVs.
Thank you.
Welcome.
So actually there is a last question from Marcel Ramirez. His microphone is not working, unfortunately, but I can ask it. So he's wondering if you have tried to remove sialic acid from the EVs to see if it's indispensable for the EV uptake and the whole process.
No, Marcel, how are you? And just for the experiment, when they use the alpha-galactose enzyme and they perform the invasion experiment, is not see if the endocytosis yet. Okay, if it depend the alpha-gal epitopes. It's a good question, Marcel. Thank you.
Okay. I don't think there are more questions. This was very interesting. Jan Lotwell just said, great talk, but he was falling asleep. It's like 1 a.m. in Sweden. So this was great. Thank you
so much. Yes, but I'd like to people, if you any doubt, if you start to collaboration, I'm so open because IVIS is a communication. It's very good. And thank you for the opportunity to share our data with everyone here.